Aloha and welcome to Botany 101. I'm Angela Nishimoto. So today we're going to go into growth, part 2, okay? And I wanted to remind you about the topics that we covered last time, as well as the topics we're going to cover today. Okay, so last time we went through growth, differentiation, and development, nutrients, vitamins and hormones, okay, plant hormones, and we started on plant movements. Today we're going to continue with plant movements and go to photoperiodism, okay, among other things, okay? Okay, so to start with, we'll start with turgor movements. Okay, I think the last thing we were talking about last time was tropisms. Okay, so we're done with that. All right. So turgor movements have to do with changes in internal water pressure. Okay, and these changes are oftentimes initiated by contact with objects that are outside of the plant, such as in bladderworts and in Venus's flytraps. Remember, these are so-called carnivorous plants, right, that actually trap insects. Look at this poor thing trapped in here. Okay, so they actually do their mechanisms, you know, the bladderwort sucking in an insect, and the Venus's flytrap closing like a trap, you know, like a bear trap on an insect by changes in internal water pressure. Okay. Also, the sensitive plant, also known locally as sleeping grass, okay? It actually has at the base of the leaflets, remember what leaflets are? Leaflets are like the little structures that make up a compound leaf. Okay, so sensitive plant or sleeping grass has these compound leaves, and at the base of the leaflets and at the base of the leaf itself, there's a structure known as a pulvinus. Okay, so what happens is that when something, an insect wants to eat this plant, or somebody touches it or steps on it, okay, the pulvinus actually makes the water pressure less, and then the leaflets fold, and the leaf itself can actually fold over, okay? A lot of people have seen that. Now there's an orchid called the catasetum, okay? Catasetums are orchids that are sort of unusual for orchids because they have unisexual flowers, okay? And also dimorphic flowers. That means the flowers of the male plant that has male flowers actually has a different structure than the female with its female flowers. Okay, this is unusual in orchids because usually in orchids you have bisexual flowers, okay? So the male flower actually has a cross trigger like this in front of where their nectar is in the back of the flower, okay? And when a bee is large enough to actually push these two triggers here, what the orchid does is it pushes the trigger, the orchid flings out its pollen onto the body of the bee, okay? So that has to do with changes in turgor pressure also, okay? Now there are also sleep movements, okay, so-called sleep movements, especially in the legume family, which are known as the Fabaceae, okay? So these includes beans and peas, also shower trees, and also koa haole. So if you happen to be wandering around at dusk or something, you might notice that the leaves or petals tend to fold, okay, sort of as though the plant is going to sleep, okay. Usually the plant will fold its leaves and petals at dusk or when the sun is going down, and then unfold them when the sun is coming up, okay, or during dawn, okay. Now another kind of movement of plants is called solar tracking. This usually occurs in leaves, although we see here some sunflowers, okay, sort of turning their faces toward the ambient light, okay. So leaves will oftentimes twist and become oriented toward the light, okay. So some call this heliotropism, but it's actually more accurately called phototorsion, because tropisms have to do with growing towards something, whereas this just involves twisting. Okay, so what we're going to do is we're going to go over and take a look at a plant over here. Okay. Okay, so this here is an aglaonema. All right, it's a very common plant used in landscaping and as a house plant in Hawaii. Okay, in the nursery trade they call them aggies, aglaonemas, and they're related to kalo, they're related to Dieffenbachia, they're related to the that 9-foot tall, plant that has that big inflorescence, the Amorphophallus titanum or the titan arum. Is related to anthuriums. It's because the flower has this kind of structure where you have a spathe, and a spadix that actually makes the inflorescence. Okay, the spathe being a leaf-like structure, which some people think of like petal-like, okay, and a spadix, which is where the flowers are. All right. So if you look at this plant here, one of the things that you'll see, okay, is that the leaves are all turned in one direction. Okay, this plant actually lives on my countertop next to my stove. Okay, and it gets a unidirectional source of light, or the light comes in from one direction. If I turn the plant to the other side, you can see that almost no leaves are facing in the opposite direction. Okay, so generally the light actually hits a plant from this area here. Now, when you look at this plant also, you might see these little dots along the stem. Okay, these are where there are calluses being formed or undifferentiated tissue, and they're forming places where roots can come out of the stem. So this plant is actually forming adventitious roots. Okay, so if we turn it back this way, you can see that the leaves are all on one side. Okay, phototorsion, okay, or turning toward the light. Okay, anyway, what we'll do now is we'll go back to the computer. Okay. Okay, now we're back again. Okay, so we also have water conservation movements. Okay, grasses have bulliform cells in their leaves. Okay, remember we saw this particular image before. Okay, so the leaves of grasses have these thin-walled cells called bulliform cells, okay, below grooves in the leaf surface. Okay, so when water is scarce or not available, the cells lose turgor, they sort of collapse, and then the leaves either roll up or fold. Okay, so then what happens is that transpiration decreases to 5 to 10% of its normal rate. Okay, so that's a one kind of water conservation movement. Okay. Now we're going to talk about taxis. Okay, these are when the entire plant or reproductive parts move. So this does not occur in angiosperms or flowering plants. Okay, what happens is that the cell or entire plant has flagella or cilia. Okay, these are like little filaments that kind of come out of the plant or the alga, okay, and helps them to move. So this requires the ability of the cell to move, and the cilia and flagella help facilitate that. Cilia will actually wave this way to actually propel the cell or push the cell in a certain direction, and flagella will actually wave around, okay, to push or propel the cell into either the direction of a stimulus or away from a stimulus, okay? Now there's different kind of taxis. For one thing, there's chemotaxis, okay? One example of this is the sperm of ferns, okay, will swim toward the female structure in a fern, okay? Because the female structure will produce chemicals that actually attract the sperm cell, okay? We also have phototaxis, and this here is a cell of a single-celled alga called Chlamydomonas. Here you can see the two flagella. Yeah, so that helps the cell propel itself through the liquid media in which it lives in. Okay, so phototaxis is movement towards or away from the light, and Chlamydomonas has an eye spot in it that actually makes it swim toward the light, because of course this is a green alga, so it does photosynthesis, so it needs light in order to live. Okay, so phototaxis is a movement of the whole organism toward the light. Okay, then we have aerotaxis. Okay, this is something that we talked about before in Engelmann's experiment, right? We had these oxygen-loving bacteria actually moving to areas along the spectrum where most photosynthesis was taking place, mostly in the blue and the red. Okay. Now we're going to shift direction a little bit and get into photoperiodism. Okay. Now photoperiodism has to do with the effect of light, or the timing of light, on flowering. Okay. So in the early 20th century, there are these American plant physiologists named Whiteman, Garner, and Henry Allard. Okay, they actually had a tobacco plant that grew 10 to 13 feet tall, a giant tobacco plant. Okay, it was a giant tobacco plant, but it didn't flower in August when normal tobacco plants flower. Okay, so what they did is they took this tobacco plant to a greenhouse, and it flowered in December during a short daytime, or also a long night time. Okay, so what they did is they took tobacco and soybeans and gave them only 7 hours of daylight. Okay, and when they did that, all the plants flowered. So they discovered that the length of night was related to the onset of flowering. So they're the people who discovered photoperiodism. Okay. Now short day plants need less than 12 to 14 hours of daylight per day in order to flower, and usually in a temperate area that means spring or fall. Okay. And some short day plants include asters, mums, dahlias, poinsettias, which of course tend to put out their bracts and their flowers during the Christmas season when you have short days, right? Also strawberries are short day plants, and also violets. Okay. There are also long day plants, okay, and these need more than 12 to 14 hours of daylight per day. Okay, and that's usually during the summertime. Okay. And these plants include beets and lettuce, potatoes, spinach, and wheat. Okay, so leafy vegetables such as lettuce and spinach actually need to be harvested in the spring. Okay, when they're first grown, when you first sow the seeds, and then grown again in the fall to avoid what they call bolting. Bolting is when they produce flowering stalks, and that's not what you want when you want a leafy crop such as lettuce or spinach. Okay. Now there are also intermediate day plants, okay, that has two barriers. They will not flower during the day, during when the day is too short, and they will not flower if the day is too long, such as this Indian grass here. Okay, that's what they call intermediate day plants. You only flower within certain kind of parameters. And then there are day neutral plants. Okay, so these will flower under any length of day, just as long as they have enough light for normal growth. And day neutral plants include garden beans, carnations, cotton, roses, sunflowers, and tomatoes. Okay? So photoperiodism is actually a tremendous way that plants have grown to actually allow their survival, because it allows for flowering, okay, fruiting and setting seeds for the time that's best suited to the plant to ensure the survival of its offspring. Okay? Now to review what we just talked about, we talked about some plant movements. Okay, we continued with that. And we talked about photoperiodism. Okay? Now we're going to move to the next section, which has to do with meiosis and alternation of generations. Okay? So plants, we have been able to see that they can do both asexual or sexual reproduction. Okay, so generally asexual reproduction happens when a plant has one parent. Okay, that comes from the root or the stem or the leaf, usually the new plant comes from. Okay, or plants can do sexual reproduction. Okay, and sexual reproduction involves sex. Okay, it involves sex cells, which is synonymous with gametes. Okay, or they're called sperm or egg. Okay, the sperm donor is usually considered male. The egg donor is generally considered to be female. Okay, so here we actually see fertilization going on. You have here a pollen grain on the stigma of the plant. Okay, and these ovules are actually sending chemical signals, chemotaxis, right? So what happens in a way, okay, because the pollen grain actually stays up here, it doesn't actually move. What actually happens is that the ovules, or the eggs inside them too, will send chemical signals, and if this is the right pollen, the pollen will germinate and will grow a pollen tube down from the stigma into the style and then into the ovary where it produces two sperms. Okay, so in this pollen tube you have two sperms. One of them will fertilize the egg, and that becomes the embryo, the new plant or the new generation. The other sperm will fertilize these two cells called polar nuclei. Okay, and the polar nuclei become endosperm. Do you remember what endosperm is? Remember, endosperm is a nutrient source for the embryo and the seed. Okay, so when the sperm and egg, unite, you have the result is a zygote, which is diploid. It has two sets of chromosomes, and it's a new combination of two parents. Okay, the pollen-donating parent and the pod parent, the one that makes the fruit, or the female parent. Okay, so what you have is a new individual with two sets of chromosomes combining traits from its male parent and its female parent, just like in animals. Okay. Now what we need before we can actually do fertilization is we need to have meiosis. Okay, meiosis leads to gametes with half the chromosome number of their parent cell. Okay, so generally all cells have two sets of chromosomes, one set from mom, one set from dad. Okay, same as in humans, same as in plants. Okay, so generally what we have is we have two sets of chromosomes. So this looks like maybe an egg nucleus and a sperm nucleus before they fuse. Okay, so they have two sets of chromosomes, one set from mom, one set from dad. Okay, so generally what happens then is that you have homologous chromosomes, okay, inside of the cells, okay, inside of the nucleus of the cells. So homologous chromosomes are chromosomes like this one, of course. The blue one is from dad, this one is from mom, okay. They'll have the same size, okay. They have the same amount of DNA, and they have the same genes on them. Genes code for proteins. Genes code for traits, okay. Also the centromere, this part of the chromosome, for homologous chromosomes are in the same place, okay. Now what would happen if the gamete chromosome numbers were not reduced? Okay, what would happen is that you'd have a doubling of chromosome numbers after every generation if chromosome numbers were not reduced during meiosis. Okay, so if you started with a cell with 16 chromosomes, okay, after 20 generations you would end up with cells that had over 8 million chromosomes. That would be unable to be handled. That wouldn't happen. Okay, so meiosis actually reduces chromosome numbers in the gametes by half. Okay. So remember that DNA doubles before mitosis. So DNA also doubles before meiosis. Okay. So what happens is that you'll have the DNA doubling so that each chromosome has two identical DNA molecules joined at the centromere. Remember what they're called? They're called sister chromatids, right? Remember the interphase and mitosis? Okay, so when does this take place? When does this DNA doubling take place? It takes place during the interphase. Remember the interphase, right? You have the interphase and you have the mitotic phase, right? The interphase is divided into three parts. You have the G1 phase or the gap 1 phase where the cell actually enlarges, right? And also makes more organelles, okay? Then you have the S phase or the synthesis phase, and it's during this phase that DNA is duplicated. When you have one chromosome, single chromosome like this, it actually doubles the DNA and protein, so you have a double chromosome, okay? And then, of course, you have the gap 2 phase, when the cell readies itself for division, okay? Now, meiosis generally takes longer than mitosis. In lilies, meiosis takes about 2 weeks, whereas mitosis takes about only 24 hours. So meiosis is much more involved. Okay, proceeds with 2 divisions, meiosis 1 and meiosis 2. And meiosis 1 is sometimes called reduction division because during meiosis 1, the chromosome numbers have been reduced by half after meiosis 1. Okay, so it starts with prophase 1. All right. It's called prophase 1 because it's a prophase of the first division in meiosis. Okay, remember that meiosis proceeds with 2 divisions. Okay, so in prophase 1s, the chromosomes condense, becoming shorter and thicker, like you see in this image here. Okay, another thing that happens is that homologous chromosomes pair up. Okay, that doesn't happen in mitosis. Okay, but homologous chromosomes will pair up. So the chromosomes that code for the same genes, for the same proteins, will pair up with each other. Okay. The nuclear membrane and also the nucleoli break up. Okay. And a special event called crossing over can occur. Okay. So that is actually exchange of DNA between homologous chromosomes. Okay, so each pair of homologous chromosomes has 4 chromatids, right? So each chromatid is a double-stranded DNA molecule and its associated proteins, okay? Another thing that happens, that can happen is recombination. So that actually allows for recombination of parental chromosomes from mom and from dad, okay? And that is a source of variability. It's sort of like exchanging a chrome grille from a red car with the identical blue car with a black grille. You still have the same car, but each will have a different combination of parts, okay? Another thing that happens during prophase 1, the spindle starts to form, okay? And before I go on, I want to tell you that these cells that meiosis is going on, these are actually pollen grains, okay, pollen grains that they're going to result in. Okay, so this is a sporocyte, okay. So the spindle forms. Now during metaphase 1, okay, the homologous chromosomes are actually lined up directly opposite each other at the equator of the cell. Okay, so here are the homologs. You can see how dense they look. Okay. And the spindle is more apparent. Okay, you can actually see a little bit of it here. In plant cells, they're a little bit hard to see, but these little streaks are actually the spindle. Do you remember what the spindle is made of? It's made up of microtubules. Do you remember what microtubules are? They're actually part of the cytoskeleton. So what they'll do is they'll assemble when mitosis and meiosis are going on to make the spindle, and what happens is that one part of the microtubule will attach to the centromere of each of the 2 homologous chromosomes on either side. Okay. Now during anaphase 1, Okay, the homologs, each consisting of two sister chromatids, will migrate to opposite poles. Okay, this is also something that does not happen during mitosis. Okay, you have these homologous chromosomes migrating to opposite poles. Okay, so after anaphase 1, all the chromosomes on this side and all the chromosomes on this side are only half the number of the chromosomes we started with. Okay, then telophase 1, okay, actually happens. Okay, you have the formation of the cell plate. You have the chromosomes kind of going into a decondensed state in some cases. Okay, so generally telophase 1 is complete when one cell becomes 2. But depending on the species, the chromosomes either revert back to interphase or they can proceed to division 2. So after meiosis 1, the cells are haploid. So these two cells are haploid. They have half the chromosome number of the cells that we started with. Okay, with one set of chromosome in each cell. Okay. Now meiosis 2 is also known as equational division. It's also known as mitotic division. It's called mitotic division because it's very similar to mitosis except that the cells are haploid. Okay. Now during prophase 2, the chromosomes condense. You can see here that the cell wall has formed between these two adjacent cells. Okay, but even though they haven't separated, I mean completely disassociated from each other. Okay. During metaphase 2, okay, the chromosomes are aligned at the equator at their centromeres. Okay. Spindle forms, you can barely see it. Okay, the spindle fiber is attached to each centromere. Now during anaphase 2, the chromosomes, the chromatids separate and migrate to opposite poles. You can see these here. Remember that these are haploid, right? And then during telophase 2, the chromosomes diffuse back into chromatin. Okay. The nuclear envelope and nucleoli begin to reappear. Okay. And the cell walls form. Okay. So what you have at the end of meiosis, okay, you started meiosis with one cell with two sets of chromosomes. At the end of meiosis, you'll have 4 daughter cells. Okay, 4 daughter cells, all of them haploid, all of them different from each other, all of them different from their parent cell. Remember in mitosis, the 2 daughter cells were identical to each other and are identical to their parent cell. Okay? Now mitosis and meiosis compared. Okay? So here we have prophase of mitosis. This is in whitefish embryo. And here's a sporocyte in lilianther. Okay, so we have prophase of meiosis 1. So one thing you see is that the chromosomes in both are condensing, but in the prophase of mitosis, you don't have the homologs pairing up, whereas in the prophase 1 of meiosis 1, the homologous chromosomes are pairing up with each other to allow crossover events. Okay, the crossover events allow for recombination of the genetic material from mom, the genetic material from dad of this particular plant. Okay. Now metaphase of mitosis, here you see the chromosomes are aligned at the center, okay, of the cell, right? Here you can see the spindle attached to the centromeres, okay? But in metaphase 1 of meiosis 1, you have the homologous chromosomes aligned at the equator, okay? So, and the spindle forming or being formed, okay, spindle being more apparent. Now, anaphase of mitosis, you have the sister chromatids actually separating from one another. Okay, sister chromatids of the chromosomes separating from one another, and now they're now actual chromosomes in their own right. Okay, whereas anaphase 1 of meiosis 1 have homologous chromosomes separating from each other. Homologous chromosomes separating from each other. Okay, so each chromosome still has 2 sister chromatids in meiosis 1, even though after anaphase of meiosis 1 and we get to telophase of mitosis and telophase of meiosis 1, okay, telophase 1 of meiosis 1, here you have the telophase of mitosis where the chromosome number is still the same in the 2 daughter cells as they were in their parent cell, whereas in telophase 1 of meiosis 1, the 2 daughter cells have half the chromosome number in each cell as in the parent cell. Okay? Now prophase of mitosis and prophase 2 of meiosis 2. Okay? You have the chromosomes condensing, okay, in prophase of mitosis. Here you have the chromosomes condensed in prophase 2 of meiosis 2, but remember, the cells are haploid, okay? So they're very similar, okay? Except that the cells in the plant cells here are haploid in the cells in the anther, okay? Metaphase of mitosis and metaphase 2 of meiosis 2, okay? Here you have the sister chromatids, actually still together, but you have the spindle attached to the centromeres of the chromosomes aligned at the equator of the cell. And here is the same thing. You have the equator of one cell with the spindle fibers attached to the centromeres of these chromosomes, okay? And this side too, so we have 2 separate cells, each of them haploid, but in both cases, you have the spindle fibers attached to the centromeres of the chromosomes aligned at the equators of the cells for both, okay? Now anaphase of mitosis and anaphase 2 of meiosis 2, okay? In both cases, you have the sister chromatids separating from one another, okay, in the whitefish embryo and here also in these future pollen grains. Okay, so you have the sister chromatids separating from each other. So each chromosome will be made of one chromatid. Okay, it's no longer called a chromatid. It's actually called a single chromosome. Okay, so very similar to each other, except that in the meiosis cell, the cells are haploid. Okay, they're haploid after meiosis 1. Now in telophase of mitosis, you have the genetic material diffusing back into chromatin. You have the reformation of the nuclear membrane and the reappearance of the nucleoli. Okay, and also the cell begins to divide, begins cytokinesis. And in telophase 2 of meiosis 2, it's the same thing. The nucleus goes back, into a nuclear form instead of being diffused in the cell. Okay. You have the reformation of the nuclear membrane, all right? You have the fact that the genetic material is going back into its diffuse state, into chromatin, and you have the formation of the cell walls, okay? So after meiosis, okay, after meiosis in plants, you started off with one cell with 2 sets of chromosomes, and after meiosis 2, you have 4 daughter cells, all different from each other, all different from their parent cell. You started with 1 cell, you ended up with 4, okay? In mitosis, you start off with 1 mother cell, you end up with 2 daughter cells, identical to their mother cell, identical to each other, okay? In meiosis, you start off with 1 mother cell, you end up with 4 daughter cells, each of them different from each other during the crossover events that happen during prophase 1 of meiosis 1. They're all different from each other. They're all different from their mother cell too because they're all haploid, okay? Now, why plants have sex, okay? So imagine a population of plants on a mountain in Hawaii. So this is the Haleakala silversword, or Jarosiphium samuchensis, okay? So here you might have a population of silver swords up on Haleakala, okay? Then what happens? Maybe the environment changes, okay? It's happy, it's successful in its habitat, but the environment changes. It gets hotter or colder. you have more or less water available to the plant. You know that more water by now can actually hurt the plant, you know, as well as not enough water. Okay, there might be more fog or more pollution. Okay, this is a shot of Hilo Bay, okay, with the fog that we have in the air right now. Okay, so the plants that exist on the mountain in a specific habitat may go extinct. Okay, they may go extinct if they're not able to adapt to change. So sexual reproduction is a key for that. Okay, what that allows is new combinations of genes from two parents, right? It also, meiosis reshuffles the gene combinations, right? You have the, for one thing, you have the crossing over events that happens during prophase 1 of meiosis 1. And also after anaphase 1, you have the different chromosomes, the parental chromosomes, going into different parts of the cells, okay, different cells. So you actually have different cells resulting, okay? So you have new combinations from the genes of two parents when fertilization happens. So this is showing a life cycle. You have a plant, okay? So a plant undergoing meiosis, okay? We eventually end up having cells that are haploid. You have a plant that has haploid cells that gives rise to gametes that are haploid. Okay, then after sperm and egg come together, you have the zygote, the new cell of the new generation that has 2 sets of chromosomes and grows up into a diploid or 2N, or an individual with 2 sets of chromosomes. Okay, so you have new combinations of genes from 2 parents. Okay, and meiosis reshuffles the gene combinations, just like we just talked about during prophase 1, when you have the crossing over events, and also the segregation that happens after anaphase 1. Okay. So another thing is that crossing over sites are not random. Okay. So here we have these doubled chromosomes. Okay. And these double chromosomes, one maybe from the mom, the pink one, and the blue one from the dad. Okay, going to stereotypes. Okay, so what they'll do is they're attracted to each other during prophase 1 of meiosis 1, and they can exchange segments from each other, okay, during crossing over. So what you end up with eventually, and after these chromosomes are separated from each other during meiosis 1, and when sister chromatids are separated from each other during meiosis 2, you will end up with 4 different chromosomes, different from each other and different from their parental cells. Okay. Now the thing is that these crossing over sites are not random. Okay. There's some things called recombination hotspots. Okay, where you have more recombination going on. So some of these recombination hotspots can have 70 times more recombination than other places that don't have these hotspots. Okay, it has to do with the actual physical structure of the chromosome. Okay, because near the centromeres, where the sister chromatids are attached, the crossing overs are reduced. So these areas that are near the centromere have hardly any crossovers ever. That hardly ever happens. Okay. Whereas in farther away from the centromere, it's more often. So they're actually able to map chromosomes, okay, map the genes on the chromosomes, and can kind of predict, depending on where the genes are and how far they're spaced from the centromeres, what percentage of crossovers will happen at that particular site. Okay? So that leads us to the conclusion that genes are not randomly placed on the chromosomes. Okay? So you have here a very nice image of chromosomes. So this is actually a diagram, and here's actually a model of a chromosome. So chromosomes are actually called chromosomes, which mean colored bodies, because what they have is they actually have these areas along them that are genes that will stain different colors depending on the stains applied to them. Okay, so you actually see here a single chromosome with the genes arrayed. So generally, the farther away from the centromere the area is, the more likely you are to have a crossover event. Okay, so crossovers are not random. Okay, so we have a couple of questions. So are the so-called housekeeping genes, or genes that kind of govern the day-to-day activities and the day-to-day lives of the plants near the centromeres, the ones that cannot be changed very often or can wreak havoc on the individual? Okay. Another question is, are genes allowing adaptations to environment in hotspots? Okay. So are the genes that allow adaptation to environments in hotspots, areas where you have lots of crossover events? And that would make sense. So they don't really know the answers for sure, but they're still investigating these questions. Okay. Now we have a little section on fish and gish molecular techniques. Okay. In the 1980s, they did something called fluorescence in situ hybridization, also known as FISH. Okay. What that is that fragments of single-stranded DNA with a gene of interest was tagged with a phosphorescent dye. Okay. And this probe was put on a slide with cells that were dividing. Okay. And these cells had also been treated and were also in single strands of DNA. Then what happened is that the DNA fragment with the gene and the probe will pair with the complementary sequence it makes on double-stranded DNA. Okay? And then they view it under a fluorescent microscope to see where the gene is on the chromosomes. Okay? So the position of the gene on the chromosome would be visible. Okay, so now here we have an image. Okay, so here's a probe with a fluorescent dye. Okay, and here's a sample DNA. Okay, so what they did is they fixed the cells, they made it permeable to allow hybridization between the probe and the DNA in the cells. Okay, so what then happens is that you have these fluorescently labeled probes. Okay, so you have hybridization, and they'll mark them, and then so you have the hybridized cells, which will fluoresce under fluorescent microscopy. Okay? So it seems very complicated, and I'm sure it is, but the explanation is pretty okay to follow. Now you have GISH, which is genomic in situ hybridization. Okay? What happens is that if you breed one plant with another, okay, you can actually see, the hybrid in their cells. Okay, you can dye the chromosomes for one species one color with fluorescent dyes, and you can dye the chromosomes from another species a different color. Okay, so what you can do is you can actually view the chromosomes during meiosis and see what they do during meiosis, where crossovers can occur or whether they even do pair up or not. Okay. So here are some micrographs of the GISH molecular technique. Okay, so you have some chromosomes that are fluorescing blue, you have some chromosomes that are fluorescing pink. Okay, so these actually come from different sources. Okay, they actually dyed one color, they dyed another with another fluorescent dye in another color. Okay, so these are actually chromosomes of different kind of wheats. Okay, different kinds of wheats. Right? Now we come to alternation of generations. Okay? Okay, so we have ploidy. That means the number of chromosomes. If you have one set of chromosomes in the cell is known as haploid, designated as X. Okay? If the cell has two sets of chromosomes, it's called diploid, designated as 2X. Okay? So here's a diploid cell with 2 chromosomes. Here's a haploid cell with 1 chromosome. Okay? Of course, cells usually have more chromosomes than that. Now, if a plant has 3 sets of chromosomes, it's called triploid. Okay? Now, in a triploid plant, since homologous chromosomes of a triploid plant cannot pair, okay, during meiosis, seeds usually are not produced. Okay, so some plants that actually are triploid are navel oranges and seedless watermelons. You notice that the watermelons and the navel oranges do not generally develop seeds. That's because the way the chromosomes behave during meiosis, and you don't have them pairing up since you have 3 sets of chromosomes. But sometimes you have plants with 4 sets of chromosomes, and these are known as tetraploids. Tetra means 4 in Greek. Okay, so potatoes, pasta wheat, okay, are some examples of some tetraploid plants that are well known. Now bread wheat is a hexaploid with 6 sets of chromosomes, and strawberries are octaploid with 8 sets of chromosomes. See? So you can actually see that plants can actually have more than 2 sets of chromosomes, and this is actually a major way that plant species have developed. Okay? Now for most animals, the gametes are the only haploid cells. But in plants and other organisms, this is not true. Okay? Because plants undergo this alternation of generations. Okay? What happens is you have a 2X sporophyte in 2N or sporophyte generation phase. Okay, so the sporophyte generation, PHYTE, means plant. Remember we talked about epiphytes, that's a plant that grows on other plants. A sporophyte is a plant that makes the spores. Okay, now an X gametophyte has 1 set of chromosomes. Okay, that's a gametophyte generation, N. Okay, that's a plant that actually makes the gametes. Okay. So here you have, let's see, the sporophyte, okay, undergoing meiosis, resulting in a spore, okay, which grows into a gametophyte plant, the plant that's haploid, haploid plant that bears the gametes, actually makes gametes through mitosis, not meiosis, okay. Then when the male and female gametes come together, you have a 2X zygote or a diploid zygote, and then the cycle continues. Sporophyte, meiosis, okay, and growth of the gametophyte, and then gametes being produced and fertilization. So you have meiosis on one side, fertilization as the event on the other side. It actually marks the end of one generation and the next. Okay, so this happens in protistans or protists, in fungi, and in plants. Okay? So there are 6 rules to alternation of generations. Okay? Generally, the first cell of the gametophyte generation is a spore. The last cell of the gametophyte generation is a gamete. Okay? The cells of a gametophyte are haploid. Okay? They have half as many chromosomes as a sporophyte generation. Okay, the first cell of a sporophyte is a zygote, a diploid zygote. The last cell is a sporocyte. Okay? And the cells of the sporophyte are diploid. They have twice as many chromosomes as the gametophyte generation. Okay? So you have a change from sporocyte, okay, the sporophyte, to gametophyte occurs from meiosis. The sporocyte undergoes meiosis to produce a haploid spores. Okay? Now the change from the gametophyte to sporophyte occurs from fertilization, which produces a diploid zygote. Okay, so here we have another alternation of generation. Okay, so here we have the haploid, okay, spores growing into a gametophyte, okay, which then makes gametes, which come together in fertilization, that produces a zygote that grows into the sporophyte in the sporocytes undergoing meiosis, and the cycle continues. Okay? Now sexual reproduction, okay, it allows for new gene combinations, new genetic combinations, okay. So you have crossing over at prophase 1, recombines maternal and paternal genes. Remember we talked about that a little bit earlier. Okay. Then you have segregation at anaphase 1, randomly assorts maternal and paternal chromosomes, which have already been recombined during crossing over at prophase 1. Okay. This is showing crossing over. Okay, so these two, here's two chromosomes here. Okay, actually exchanging gene fragments. Okay, that's actually happening here too. We saw this particular image before, where the maternal chromosome and the paternal chromosome touch, are attracted to each other during prophase 1. Okay, they can touch each other at various places and exchange fragments, you know, toward the ends, especially not toward the centromeres, but more toward the ends of the chromosomes. It allows for recombining of parental chromosomes. Okay, so then at the end of meiosis 2, when you started off with 2, okay, you'll have 4, all of them haploid. Okay, so it allows for new gene combinations. Okay, so then fertilization then restores the diploid number, and you end up with a unique combination of genes. Okay, so every individual that results from sexual reproduction is unique, completely unique. Okay, that includes humans as well as other animals as well as plants. Okay, so you saw the seedlings in that overhead or that slide before. All those seedlings are different from each other. They're all different from each other genetically, even if they had the same mother and father. It's sort of like you and your siblings. You and your siblings might come from the same parents, the same mother, the same father, but each of you, unless you're identical twins, will look different from each other. You'll probably look a little bit different from your parents. You might resemble other family members. So sexual reproduction allows new combinations of genes to make every individual unique, completely unique. So the individuals that live now, once they pass, whether they're plants or animals, it'll never come again the same way. So you yourself are unique, okay, so am I, okay? Okay, let's see. So let's summarize, okay, and review, okay? So we talked about meiosis, the various stages. We contrasted that with mitosis, the various stages, prophase, metaphase, anaphase, and telophase, okay? And we talked about alternation of generations, okay? All right, let's see, that's all we have time for today. Yeah, that's all we actually have for you today, all right? So hoping that you enjoyed this lecture. Yeah, I enjoyed it too. Okay, and we'll see you next time. Aloha.